introducing Ron Firestein, who's really been an amazing partner, for us at the CBTN in Australia. In part because as we're generating all these large-scale data sets, from patient-derived samples, the bridge from that data into therapeutic context always gets interleaved with functional assays. And I think Ron and his team have probably done more on behalf of the CBTN's functional assay context, innovated with us and setting the stage for multimodal data integration that includes functional assays in ways that's unprecedented. And with that, I'll turn it over to Ron.
But thank you to the organizers for inviting me today. And I'll be telling you about our program and some of our efforts on mapping genetic dependencies at a multi-omic scale. So we all know that over the past decades, there's been large-scale efforts that have provided a deep understanding of the molecular landscapes of pediatric brain cancers. And these efforts led by groups such as CBTN, DKFZ, St. Jude's, and others have really given us a treasure trove of information regarding the different molecular alterations that exist at the DNA, RNA, protein level, epigenetic level, and so forth. And while this really provides a treasure trove of information, we still don't know which of these alterations are actually functionally meaningful. So in order to ascribe function, what we really need to do is integrate these molecular features with functional approaches. And that includes using clinically relevant patient models, such as cell lines, organoids, xenografts, and each of these have their own functions and strengths and weaknesses. And then applying functional interrogation approaches using either chemical drug libraries or genetic perturbants such as CRISPR. And the combination of these, or the integration of these approaches, is really the way that we've approached ascribing function to identifying new targets for pediatric brain cancer. So this type of approach is certainly not new. Many of you are familiar with efforts of the Broad Institute or the Sanger cell line projects, and they've applied this kind of approach really at high scale for adult cancers. For those of you familiar with DepMap, you'll know that there have been over 1,000 adult cancer cell lines that have been profiled using these kinds of technologies, and this has really provided some exciting new targets and biomarkers. But at the same time, if we look at what's been done in pediatric cancers, there's really been, at a log scale, less numbers of cell lines that have been profiled. So if you look in DepMap, there's probably around 100 or so cell lines that have been subjected to whole genome CRISPR screens and incorporated in these efforts. So we saw this as an opportunity to really expand our knowledge in pediatric cancers, in particular brain cancers. So our program called the Childhood Cancer Model Atlas really has three main aims. The first is to develop childhood cancer cell line models, and that includes both in-house development. Our program's based in Melbourne, Australia, and we also develop models from our hospitals, but also from sites around the world, and we have 40 or 50 different partner sites that send us either existing models or samples from which we do that. And then these models are subjected to both molecular feature analysis as well as drug and genetic screens. And the last aspect of this is we integrate the data and actually share this publicly with a data dashboard and a portal. And this is just a snapshot of our dashboard, and you can see over the past year and a half, we've added an analytical portal that allows anyone to go and query the different molecular features by gene or by type of tumor. You can see that as of now, we have the program started in 2017, as you can see. From 0 cell lines, we've brought in over 600 samples over the last 7 years, of which about 440 we've been able to establish active cell lines that have gone through this pipeline of whole genome sequencing, DNA methylation arrays, RNA sequencing, total and phosphoproteomics, as well as CRISPR and drug screens. And you can see the current numbers, which are updated monthly. Importantly for this group, you can see here color-coded in red that the majority of the cell lines that we have are actually pediatric CNS lines. Our largest cohorts are diffuse midline gliomas and hemispheric high-grade gliomas, but we also have a good cohort of rare or ultra-rare types of CNS cancers like ETMRs, CPC, ependymomas, and going through interrogating those and performing some CRISPR screens to identify new targets there. So I'd like to just give you a vignette for the rest of the talk about how we're using this kind of data sets to really accelerate the identification of targets, but also in the context of precision medicine, be able to match targets to particular patient or tumor types. So we all know that these models are only beneficial if they recapitulate the disease entity. So putting these models in the context of the WHO classification of pediatric high-grade gliomas, it's important to recognize that these gliomas are not a single entity. They're now classified based on molecular features, including histone mutations or signaling pathways that are altered in these cancers. And this gives us an excellent opportunity to match our models to these molecular classifier and then begin to understand what are some of the vulnerabilities that may be unique to each one, or we might uncover that the fact that they all respond to the same target. So that's one of the first questions we did, and using these cell line models, we are in fact able to match our cell lines at a deep level across these different types of histone mutant pediatric high-grade gliomas and begin to ask what are some of the distinct differences between pediatric and adult across age ranges based on tumor location or lineage, as well as based on different molecular subtypes of the histone mutations. Two talented postdocs in the lab, Paul Daniel and Claire Soon, really started with the question of taking all of the CRISPR data that we have from these cell lines, and this is data from over 100 cell lines. Most of these are pediatric high-grade gliomas and a cohort of adult high-grade gliomas, and asking what are the differences between pediatric and high-grade gliomas. And what you see here in this scatter plot are genetic vulnerabilities. Color-coded in green are genes that are specifically or uniquely important in the context of pediatric gliomas. And I think encouragingly, you can see there's a number of targets here that are actually therapeutically relevant, such as the PI3 kinase, signaling pathway, PIK3CA, FGFR, EZH2, and MCL1. It's important to recognize that a critical part of precision medicine is identifying the patient population that is going to really respond to these kinds of targeted therapies. And this is a question that we've been addressing using machine learning approaches. And Claire and Paul, what they did is they took these genetic vulnerabilities that are occurring in pediatric gliomas, and they asked, are there molecular features that correlate to these genetic dependencies? And do these molecular features, in fact, are they the same or different between pediatric and adult cancers? So you can see here, if you focus on the dots color-coded in yellow, these are genetic dependencies that occur both in, that have molecular features that correlate to them in both adult and pediatric cancers. And essentially what we find is that about half of these genes do in fact have unique molecular features that correlate to response in pediatric cancers. And those are color-coded here in yellow. And focusing on MCL1, I'll tell you a little bit about this gene. We were particularly interested in MCL1 because it was in fact the single most unique dependency in pediatric cancer. So you can see here that MCL1 in this volcano plot really bubbles up as an important genetic feature in pediatric hydrogliomas compared to adult. And you can see in the plot on the right that in fact MCL1 is an important vulnerability in the majority of pediatric hydrogliomas, but not in adult. And this is not correlated, this is not related to MCL1 expression itself. We looked at both RNA as well as protein levels of MCL1. And overall, MCL1 is equivalently expressed in both pediatric and adult glioma. So we have then asked what are the molecular features that correlate to this response in pediatric gliomas? And what we did is we applied a machine learning model called Random Forest that predicts response across these different molecular features that we input into the model. And what we found is that there was, in fact, one feature that ranked very highly in the top 5, and that's a methylation site at the BCL201 locus. We then asked whether how this correlates to the other sites in the BCL201 locus. So if you turn your attention to the lollipop plot on the right, you can see that, in fact, this particular site, 298, it maps within a cluster of methylation sites, CPG sites that are in the exon 2, intron 2 boundary. And in fact, you can see that a correlation matrix analysis really showed that there is a cluster of about 10 methylation sites that are present in this boundary that are very tightly linked to one another, indicating that there is a methylation pattern that correlates to this genetic vulnerability. And in fact, we were able to extend this and show that these BCL201 methylation sites, they actually correlate very nicely to response to MCL1 inhibitors. So in this case, what we've done is we looked at our drug screening data across 38 pediatric glioma cell lines that were subjected to the inhibitor. And you can see the waterfall plot on the right. What we can see is that the cluster 1 CPG sites, color-coded in purple, in fact, are the ones that are most tightly correlated to response to the MCL1 inhibitor. Interestingly, the DepMap group also identified MCL1 genetic dependency as a unique feature of pediatric cancers in the context of extracranial pediatric cancers. So as some of you might know, there's a cohort of around 100 cell lines in the pediatric cancer DepMap site that are already publicly available, but only one of these are gliomas. So what we're focusing here is mostly on neuroblastomas and different types of sarcomas, but nevertheless, you can see that about half of these extracranial pediatric cancers are in fact dependent on MCL1. But at the time, DepMap did not really investigate any biomarkers that correlate to this. So we decided to look at the DepMap data as well. And in fact, what we found is that these methylation sites are highly predictive of MCL1 dependency, not just in CNS cancers, but also in other extracranial pediatric cancers. And you can see here on these box plots, looking at the correlation of the methylation sites that are in cluster 1. So the way DepMap performs their methylation data is using bisulfite sequencing. So we had to actually identify, we could only identify 2 cluster 1 sites within their data, but nevertheless, these 2 cluster 1 sites are highly enriched in pediatric cancers compared to adult cancer cell lines. So this provides us additional data that this site is in fact important in other pediatric cancers as well. So we've looked at tumors. So previously, up until now, we've shown you only cell lines, but this is data from different types of pediatric tumors. And you can see that overall, many pediatric tumor types, both CNS cancers and extracranial cancers, have either high methylation at the BCL201 locus or have a binary pattern. And lastly, you can see that the drug response does also correlate in these these other tumor types in ATRTs, osteosarcomas, and ependymomas. In each case, the methylation site correlates to response to the MCL1 inhibitor. Lastly, what we've shown as well is that this also extends in vivo. So these are ATRT models that either have the methylation site on or off. And very briefly, you can see that when we implant these tumors in the brain and knock out MCL1 and other genes, you can see that MCL1 dependency is a unique feature of the BT12 where harbors the BCL201 methylation compared to BT16. At the end of the day, this really does correlate to BCL201 expression. We show that if you restore BCL201 gene expression by exogenous introduction of BCL201 to these methylated cancers, you do in fact generate resistance to the inhibitor. So the methylation clearly does basically suppress the expression of BCL201. Now, on the flip side, we also have to consider what is the methylation status in the normal tissue as well, because if BCL201 is also methylated in normal tissue, we would expect a lot of toxicity effects. But strikingly, what we found is looking at least across that normal brain tissue methylation rates, that in fact, at least in brain, and we haven't been able to do this extensively in other tissues as well, but at least in brain, we see that the BCL201 cluster methylation is actually lower in the non-malignant pediatric brain compared to adult. And that's in contrast with global DNA methylation, which is increased. So we think, at least from a biomarker perspective, we might have a good therapeutic window. So just to put it all together and potential translation into the clinic, I'll just preface by saying that current MCL1 inhibitors have generally not done very well in the adult space due to cardiotoxicity, but that doesn't mean that in the pediatric space they might not perform well, or there might be new inhibitors coming about. But our current model really suggests that in the context of pediatric brain cancers, we have a preponderance of BCL201 methylation that represses BCL201 expression and renders these pediatric gliomas highly sensitive to MCL1 inhibition, as opposed to the normal brain tissue, which has a higher level of BCL201, and we expect them to be less toxicity. And this is in contrast with adult cancers, which have a higher level of BCL201 and hence resistance to the MCL1 inhibitors. Lastly, from a biomarker perspective, we know that methylation arrays are now becoming standard practice in the diagnosis of pediatric brain cancers, and that I think really enables an efficient method for us to look at BCL201 methylation, and in the sense that the diagnostic assay is already out there and can give clinicians an indication of BCL201 status and potential response to MCL1 inhibitor. So I'll just conclude here. I'd like to especially thank the CBTN group, Mateus, Kapitra, and others who through the years have enabled us to build this cell line cohort. I think about 70 or 80 of our models are actually from the CBTN investigators who generously contributed, as well as obviously the patients and families, their generosity, our funding partners, and the team in Australia and beyond.
Very good now. Thanks so much, Ron. Hopefully you can see that the germline predisposition is sort of the other side of CRISPR screening in some respects in terms of identifying targets and opportunities. And as Ron mentioned, CBTN has been thrilled to partner with Ron in this context.